I'm sure you're all wondering how my finger's doing. At the minute, it's still not fully healed, but it's definitely getting there. And the reason that it's not fully healed, I'm sure you're aware, is that mitosis takes its time using cellular division, so it's obviously not going to heal all in one go. But the question is, if a cell is dividing, then how is the genetic information that is stored being reproduced to each of these cells? In the future we'll learn about mitosis, but for now we'll learn about exactly how DNA replicates. Let me explain. The important thing to remember about DNA is that it is, of course, a double helix, meaning that there are two strands to this structure. In this process, one of these strands will remain exactly the same, and the other one will also remain exactly the same, but it will no longer be part of the same strand. Our DNA helix will eventually split in two, but each of these strands will form a new strand from this original DNA double helix. The two new daughter double helixes will have one strand that is identical to the original and one strand that has formed from the cytoplasm of the cell. And we will have two instances of this occur. We describe this as semiconservative, this whole procedure, meaning that one of these strands will remain the same and the other one will be a brand new strand. This allows our bodies to maintain some form of continuity throughout the generations of DNA. Now, as you're aware, the double helix of a DNA strand is a curled structure. So initially what happens in this procedure is the DNA is effectively unravelled and a little enzyme called DNA helicase goes along and effectively breaks apart the hydrogen bonds between the nucleotides of the DNA molecule. We refer to this process as unzipping, where the single double helix will form two separate strands. What happens then is any free-floating nucleotides which naturally occur in the cell will be attracted to the exposed length of each of these strands, and naturally a condensation reaction occurs using the enzyme DNA polymerase, which also helps build the sugar phosphate backbone along these nucleotides to form one strong strand, which then bonds to the original strand. So there you can see we have one initial strand and one newly formed strand in two separate cases, because there are two separate strands that are formed from the original helix. This model is described as the Watson and Crick model. This model helps us to understand that DNA is not just being cloned and pulled apart and cloned, it's actually using itself as a template and forming new strands in a semi-conservative fashion. There's an experiment we can do to prove this model by placing heavy DNA using a heavier nitrogen isotope, and then we can suspend that DNA solution in water. Where the DNA's strip forms will tell us how heavy the DNA is at any given point. We can then move the heavy DNA to an atmosphere of regular nitrogen isotope concentration, nitrogen-14 usually, but we will see that when following generations are formed, you will get a brand new layer of DNA formed. And over time you see that the areas that are occupied by these suspended DNA are indicative of a situation where at least one of the strands is heavier.